cell-mediated immunity. Cell-mediated immunity is targeted to killing intracellular organisms, intracellular bacteria and viruses, complex structures such as parasites, destruction of tumor cells, rejection of graft tissue, a delayed type hypersensitivity reaction after contact with certain antigens such as poison ivy and certain topically applied drugs. Cell-mediated immunity involves three major defense strategies. Number one, activating antigen-specific cytotoxic T lymphocytes. Number two, activating macrophage natural killer cells and antibody-dependent cytotoxicity. Number three, stimulating cells to secrete a variety of cytokines. First, activating antigen-specific cytotoxic T lymphocytes. T helper 1 lymphocytes recognize antigens represented by macrophages and function primarily to activate and heighten cell-mediated immunity by producing cytokines such as interleukin-2, interferon gamma, tumor necrosis factor beta. These cytokines have many functions, include enable TCD8 lymphocytes to proliferate and differentiate into cytotoxic T lymphocytes capable of destroying infected host cells and mutant cells. Activate natural killer cells. Activate macrophages, enabling them to destroy intracellular pathogens. Stimulate the production of opsonizing and complement activating antibodies for enhanced attachment during phagocytosis. Activate neutrophils. Stimulate increased production of monocytes in the bone marrow. Function as chemoattractants for phagocytes. Here, the activation of CD8 T cells presented by antigen-presenting cells present antigen on MHC class II molecules, to helper T cells that secrete lymphokine secretion, including interleukin-2, that aids in T cell proliferation and activation of CD8 T cells to cytotoxic T cells. Also, cytotoxic T cells can recognize the antigen on the MHC class I molecules, the same antigen that has been activated by the T helper 1 cell. Okay? Here the cytotoxic T lymphocyte activity that begins with recognition of CD8 T cells of antigen and MHC class I molecules represented by antigen-presenting cells, then the process of activation by T helper 1 cells, secretion of interleukin-2, then the activated cytotoxic T cells begin its lysis activity on the other infected cells. Cytotoxic T cells kill virally infected target cells by direct cell lysis, otherwise known as apoptosis or programmed cell death. For lysis to occur, the target cells must carry both the same viral antigen and the same MHC class I antigen as the cells that originally induced the proliferation of T cytotoxic cells. The T cytotoxic cells recognize antigen in conjunction with MHC class I molecule, are stimulated by T helper 1 cells, and then turn cytotoxic. The MHC class 1 antigens appear to be ideal recognition antigens triggering cytotoxic T cells because they are present on almost all cells. A wide variety of cells can become virally infected or turn malignant. Hence, it is essential that cytotoxic T cells have a wide repertoire of target cells against which they can mediate their killing action. These cytotoxic cells may thus be the major effector pathway of cell-mediated immunity to certain viruses. This happens just after a few hours of viruses infecting cells before the viruses can replicate. The mechanism of cell lysis by cytotoxic T cells and natural killer cells inducing apoptosis by at least two different pathways. The first and most effective pathway involves intracellular granules, where cytotoxic T cells contain granules composed of proteoglycans to which chemokines are complexed. These granules hold pore-forming proteins called perforins and proteolytic enzymes called granzymes in a protected state. The second pathway by which cytotoxic T cells can trigger apoptosis of the infected cells is through Vas-L-Vas interactions. Cytotoxic T cells have a receptor called Vas-L that can interact with Vas molecules found on the surface of most cell types. This Vas-L-Vas interaction triggers an intracellular transduction that activates the caspase enzyme and leads to destruction of the cytoskeleton and chromosome in the infected cell. Here, the first and most effective pathway that involves intracellular granules, the cytotoxic T cells become closer to the target cell and defined it through the MHC class 1 molecule, so it began to secrete its granules on its cell surface and pores formed on the cell membrane of the infected cells, and the cell began to program cell death. The second pathway by which cytotoxic T cells can trigger apoptosis through Vas-L-Vas interactions that triggers also the cell lysis. Cytotoxic T cells are not destroyed in these reactions. They can function over and over again to destroy more virus-infected cells. Death by apoptosis does not result in release of cellular content such as inflammatory mediators or viruses as does cell lysis. Instead, the cell breaks into fragments that are subsequently removed by phagocytes. This reduces inflammation and also prevents the release of viruses and their spread into uninfected cells. In addition, the activated enzymes that degrade host DNA can also destroy microbial DNA and thus kill infectious microbes within the cell. Mechanisms of evading cell-mediated immunity. How can the microbes escape from cell-mediated immunity? A high rate of mutation in some viruses like HIV and hepatitis C virus, it changes the amino acid sequence and therefore the shape of the K epitopes. Cytotoxic T cells with T cell receptors made against the cellular strains of these viruses may no longer bind to and lyse cells infected with mutated strains. Many viruses such as the cytomegalovirus and adenoviruses and some tumor cells can block the formation of MHC class 1 molecules by the infected cells. As a result, the cytotoxic T cells are no longer able to recognize and that the cell is infected and cannot kill it. Epstein-Barr virus down-regulates several host proteins involved in attaching viral epitopes to MHC class I molecules and displaying them on host cell surface. Adenovirus and EBV virus code for proteins that block apoptosis of viral infected cells. Activating macrophages, natural killer cells, and antibody-dependent cytotoxicity, the second mission of cell-mediated immunity. Activated macrophages. Activated macrophages are the effector cells in the cell-mediated immune response to virus microorganisms. In this effector pathway, the macrophage ingests the microorganism, processes the antigen, and then presents the antigen on its surface in association with MHC class II molecules to helper T lymphocytes. This triggers the T helper 1 cells to secrete several cytokines including interferon gamma. They bind to specific receptors on the macrophage coding its activation. Activation of macrophages leads to increased production of toxic oxygen radicals, nitric oxide, and hydrolytic lysosomal enzymes, enabling the killing of microbes within their phagolysosomes. Causes the macrophages to secrete cytokines such as tumor necrosis factor alpha, interleukin 1, interleukin 12. Tumor necrosis factor alpha and interleukin 1 promote inflammation through circuit phagocytic leukocytes. Interleukin 12 enables naive T4 lymphocytes to differentiate into T helper 1 cells. Also, activated macrophages leads to increases the production of B7 co-stimulator molecules and MHC class I molecules by macrophages for increased T lymphocyte activation. Also, interferon gamma produced by T helper 1 cells increases the production of opsonizing and complement activating immunoglobulin G to promote enhanced attachment of microbes to phagocytes. Here we see the antigen-presenting cell presenting antigen molecules on MHC class II molecules to CD4 T cells that begin to be activated to T helper 1 cells, starting secreting chemokines interferon gamma that binds to specific receptors on the antigen-presenting cells. Thus, it becomes activated and begin to cell lysis for the microbes. So it is a circle. The second killer cells are the natural killer cells. Natural killer cells are large granular lymphocytes that carry the cell surface markers CD56 and CD2. These cells appear to have both anti-tumor and antimicrobial activity in vivo. Cytokines such as interleukin 2 and interferon gamma produced by T helper 1 lymphocytes activate also natural killer cells. Natural killer cells also secrete the cytokine interleukin 1 in response to interleukin 2. They become super killers. Natural killer cells attach to their target cells by a receptor. Once the natural killer cells are activated, they cause lysis and apoptosis of the target cell by mechanisms similar to those described for cytotoxic T cells above. The main target of natural killer cells are tumor cells and certain virally infected cells. Natural killer cells appear to use a dual receptor system in determining whether to kill or not kill human cells. The first receptor, called the killer activating receptor, can bind to a number of different molecules usually present on nucleated human cells, and this sends a positive signal which enables the natural killer cell to kill the cell to which it has bound unless the second receptor cancels that signal. This second receptor, called the killer inhibitory receptor, recognizes MHC class I molecules, which are also usually present in all nucleated human cells. If MHC class I molecules are expressed on the cell, the killer inhibitory receptor sends a negative signal that overrides the kill signal and prevents the natural killer cell from killing that cell. Viruses often suppress class I MHC expression in cells they infect. The virus-infected cell therefore becomes susceptible to killing by natural killer cells. Tumor cells have reduced or no class I MHC expression. They too become susceptible to killing by natural killer cells. The cytomegalovirus evades the immune system because it can trigger its host cell to produce altered MHC class I molecules that are unable to bind viral epitopes and therefore are not recognized by cytotoxic T cells. However, natural killer cells are also unable to kill this infected cell because it is still displaying MHC class I molecules on its surface naturally. Antibody-dependent cell-mediated cytotoxicity. Natural killer cells are capable of antibody-dependent cellular cytotoxicity, ADCC. When natural killer cells are carrying out ADCC, they are sometimes also referred to as killer cells, key cells. Killer cells have receptors on their surface for the Fc portion of immunoglobulin G. The antibody, usually specific immunoglobulin G against foreign cells antigen, binds to an Fc receptor on the killer cell and to an antibody combining site via the Fab portion on the target cell. The antibody acts like a bridge between the specific antigen-bearing target cell and the killer cell. The killer cell then releases perforin proteolytic enzymes called granzymes and chemokines that cause cell apoptosis and cell lysis, similar to the mechanism of killing described for cytotoxic T cells. Here we see the antibody-dependent cell-mediated cytotoxicity. The activated killer cells have Fc receptors that can bind immunoglobulin G antibody that also can bind by its Fab portion the antigens on presenting on target cells, the process like ozonization. So the activated killer cells use the antibody as a bridge, then begin to kill the target cell. Monocytes, neutrophils, and eosinophils can also participate in ADCC. Eosinophils, for example, are effective killers of antibody-coated schistosomes, the larval forms of schistosomes. The mechanism of killing involves binding of the eosinophils to the larva via an antibody molecule. The eosinophil releases basic proteins and other molecules from its granules, leading to formation of pores in the target cell membrane. The organism dies because of the leaky membrane. Eosinophils can be activated by both monokines and certain lymphokines. Secretion of cytokines. Secretion of cytokines during cell-mediated immunity. Cytokines are soluble substances produced by a variety of cell types that amplify and regulate a range of immune responses. Some cytokines are crucial in cell-mediated immune reactions, whereas others play a critical role in antibody responses. A variety of cytokines affect cellular immunity by mediating the innate immune response, regulating hematopoiesis, directly influencing cell-mediated immunity. Cytokines that affect the inflammatory response generally upregulate innate immunity. These cytokines also play a role in mediating resistance to viral infections and coding the cardinal signs of inflammation, including pain, redness, swelling, and heat. Included in this group are the type 1 interferons alpha and beta, tumor necrosis factor, and interleukin 1, interleukin 6, and interleukin 8. Several cytokines play a major role in hematopoiesis in the bone marrow. The best characterized of these are granulocyte monocyte colony stimulating factor, granulocyte colony stimulating factor, stem cell factor, interleukin 3, interleukin 7, and interleukin 9. Some of these cytokines are being used to treat patients that have deficiencies in hematopoiesis, such as cancer patients who are bone marrow suppressed due to anti-cancer therapy. Specific cellular immune responses are mediated by T lymphocytes. Activation of T cells requires the presence of antigen. Cytokines serve as a second signal to drive the growth and differentiation of antigen-activated lymphocytes. Some cytokines act predominantly on T cells, others both on T and B cells. Interleukin 2 is produced by CD4 T helper cells. Interleukin 2 is the primary growth and differentiation factor for T cells. Interleukin 2 causes antigen-primed T helper cells to proliferate. In addition, it causes antigen-primed cytotoxic T cells to proliferate and become aggressively cytotoxic. So it is very important in cell-mediated immunity. Secretion of cytokines. I hope you enjoy listening this lesson. Thank you all.